So the other thing that I wanted to go over today was how to do a monohybrid cross, which you may or may not be familiar with. The first thing, all of this Mendelian genetics stuff is based off of probability. So I just wanted to quickly go over probability. There's like a little formula at the top of the page, this one right here, where it says number of ways an outcome can occur. Outcome can occur divided by the total possible number of outcomes. Now, this isn't a formula that you will be knowingly performing. It's just one that's kind of a little bit obvious, but just so you know where these numbers are coming from. So it's basically saying 2 out of 4 can be tall, or 3 out of 4 will be homozygous recessive, or 1 out of 4 will be heterozygous. That's what it means by probability. The number of ways that this outcome will occur, whatever the outcome we're looking for, divided by the total number of outcomes, which right now for monohybrid crosses, it's going to be 4 possibilities. That'll be your number every time, okay? Most importantly, you guys can see the star that I put. This probability can be expressed 3 ways: a fraction, so for example, 1 out of 4. Can be a decimal, which would be 0.25, or a percentage, 25%, okay? So it'll either be a fraction, a decimal, or a percentage. Generally, on the diploma, they're going to be asking for the decimal. That's kind of the majority, not all the time, but for the most part, they're going to be asking for you to give it as a decimal, okay? Now, to figure out the probability, we use Punnett squares, which look very much like a tic-tac-toe board. So you draw a square, put a little cross in the middle, you got your little tic-tac-toe board. When we do Punnett squares, we are always writing the gametes along the top or the side, generally the left-hand side. And the combinations are written inside, and those are the numbers that are going to be put in our probability, okay? So the example that I've given to you on your notes says, example, a homozygous dominant plant crossed, so we use an X for that, crossed with a heterozygous plant. And it says predict the genotype and phenotype ratios. So that's just what the example on your page says. I know I wrote it a little bit messy, but that's totally cool. So first thing we need to do is figure out what the genotypes of the parents are that we're going to be crossing. Homozygous dominant means both capital letters. We are talking about tallness. Tall is dominant. So to me, big T means tall, little t means short. Again, if you wanted to choose different letters, that's totally okay, but that's just generally what we do. I always like to write a little key because we're going to be getting to more complicated crosses, and a key is going to be helpful. So although this is kind of a little bit obvious, it's just a good habit for you guys to get into. So the parents that we have are big T, homozygous dominant, crossed with big T, little t, a heterozygous plant, okay? So hopefully you see that. Homozygous dominant, big T, that's parent number 1, crossed with a heterozygous plant, big T, little t, that's parent number 2. When we say that we write the gametes along the top and the side, and I'm just going to draw a bigger one here for us. Here's our first parent. The possible gametes, they're either going to give us a gamete with a big T or a gamete with a big T. Those are the possibilities. They're going to give us a big T either way. So for organism 1, we would write those possibilities along the side, okay? Organism 2, the possible gametes are big T, or they could give a little t. So it's either or. So we would write those along the top. If you wrote the organisms backwards and you wrote big T, little t on the side, big T on the top, going to give you the same answer. It really doesn't matter. Now we're ready to fill in the Punnett square. So either you start at the top or the side and you start to fill it in. So if we start with this guy right here, he's going to be giving a big T no matter what. this guy here is going to be giving a big T and a big T, no matter what. Then we start to work our way down. This guy is going to give a big T or big T, no matter what. This guy is going to give little t and little t. Now you can see we have 4 combinations written inside of our Punnett square. Okay, those are the possibilities. So when we write the probabilities, our genotype and phenotype ratios, here's our genotype ratios, the possibilities, big T, which I've written over here. There is a possibility of 2 out of the 4, which means 50%. Or literally writing it as a percent, either decimal or percent, okay? The other possibility that we can see is big T, little t. And those are the ones that are on the right that I've circled in blue. Probability of those occurring, 2 out of 4, or 0.5, or 50%. That's our genotype ratio. That's our probability. So being homozygous dominant or heterozygous. For the phenotype ratio, well, they're all going to be tall. There are no little t's, so we would say that 4 out of 4, or 1, or 100%, all tall, okay? So that's just a general, straightforward example. I will go through a whole bunch of these examples for you from the workbook if you guys are struggling with any of that. Now on the bottom of the page, I've written something called a test cross, and this is something that I find kids sometimes struggle with, but I'm going to do my best to explain it because it's actually super straightforward and super easy to understand, okay? So test cross. And you will see this 100% show up on your diploma. A test cross is basically just using a homozygous recessive organism to find an unknown dominant phenotype. Okay, so basically it just means if you have a tall plant and we know that tall is dominant, how do you know if it's homozygous dominant or heterozygous? How can you determine it? Because without doing genetic testing, you would have no idea what the genotype is. You only know that it's dominant. The other way that we express that, and you'll see this later on in the unit, sometimes we express it like this. We know that it has at least one dominant allele, but we have no idea what the second one is. We don't know if it's a dominant or recessive allele. There's an easy way for you to determine if something is homozygous or heterozygous. You use a homozygous recessive, because that is the only thing that you 100% know the genotype. The only way you can get a recessive phenotype is if it's homozygous recessive. So you cross it with a homozygous recessive organism. There are 2 outcomes. If the organism that's unknown is homozygous dominant, you would perform a cross, and this is what the outcome would be. Ooh, that is a very tall, narrow Punnett square. That's okay. So this is the test that you know you're taking your homozygous recessive. If the organism is homozygous dominant, you will see that every offspring comes out as having a dominant phenotype. So if it was homozygous recessive, offspring will always show dominant phenotype. You will never, ever, 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 ever get a recessive phenotype offspring. So you'll never have a short offspring, okay? So never produce recessive phenotype. So you could do it 1 time, 1,000 times, 500 million times. You will never get a recessive phenotype. That seems super straightforward. That makes sense. The other case is if your mystery or unknown genotype was heterozygous. Now, this one is a little bit different. If you take your Punnett square, which this one looks a lot better, here's your test cross. You're taking your homozygous recessive organism, and say this mystery phenotype or genotype was heterozygous. If you do this cross, big T, little t, big T, little t, little t, little t, little t, little t. The genotype ratios out of this is that half the offspring will be dominant, have a dominant phenotype. Half of the offspring will be recessive. So this is the only way that you can ever get a recessive phenotype child, is if that parent is heterozygous. So you can see, based off the outcome of this test cross, you can figure out if you cross it a whole bunch of times and even one of them comes out with a recessive phenotype, you know that the unknown phenotype or unknown genotype of the parent is heterozygous, okay? Super, super straightforward. I'm going to end this video. I'm going to start a new one, and I'm just going to go through some examples from the workbook so that you guys can feel some confidence. If you feel like you totally understand this, you don't need to watch the next video. You can just start practicing on your own. But please, guys, this is a little bit more independent. This is on you guys. If you don't do the work, you're going to struggle, and I'm not there to push you, okay? So please just use the time that you have wisely. Again, email me if you have questions. I'll do my best to answer them, okay?